complex process in which a ribosome. Translation is a complex process in which a ribosome uses the coded information found in messenger RNA, or mRNA, to build a functional protein. When you have completed this exercise, you should understand the structure and function of the ribosome, understand the function of charged tRNAs, understand the three distinct steps of translation: initiation, elongation, and termination, and understand the functions of translation factors in each of these steps. The genetic information necessary to build proteins is contained within an organism's genome. To create a protein, genes are first transcribed to create messenger RNA. The mRNA is then read by a large structure called the ribosome, which links amino acids together to form the protein encoded by the mRNA. The ribosome cannot accomplish this feat unaided, however. Besides the mRNA and a ribosome, translation also requires amino acid carriers called transfer RNAs and a large number of initiation, elongation, and release factors. Finally, ribosomes cannot be reused in a cell without the action of a recycling factor. The ribosome is a large macromolecular structure that performs protein synthesis in cells. The ribosome links together the appropriate amino acids to form the polypeptide chain encoded by the gene. The ribosome consists of two subunits, the small subunit and the large subunit, each consisting of both ribosomal RNA and protein. In contrast with many enzymes you have studied, over half of the ribosome is composed of RNA, with protein making up a smaller portion of the mass of this enzyme. Ribosomes are similar in structure in prokaryotes and eukaryotes, although the overall size of the ribosome is smaller in prokaryotes. Ribosomal subunits are named by their Svedberg coefficients, a measure of their sedimentation rate when subjected to centrifugal force. Ribosomal subunits assemble into a full ribosome on the mRNA, disassemble when the protein has been synthesized, and assemble once again to start another round of protein synthesis. This process of repeated assembly and disassembly of ribosomal subunits is known as the ribosome cycle. The ribosome reads the mRNA 3 bases at a time. Note that, while spaces have been added to aid in visualization, these triplet codons are immediately adjacent to one another on the mRNA. The triplet codons are matched within the ribosome to specialized RNA molecules that carry amino acids. These amino acid carrying RNAs are called transfer RNAs, or tRNAs, for they are adapter molecules that transfer genetic information to amino acid sequence. tRNAs contain anticodons that are complementary to, and thus bind, the codons on the mRNA. The ribosome is uniquely designed to bring mRNA and tRNAs together. The mRNA is threaded through the small subunit, while the large and small subunits of the ribosome fit together to make tRNA binding pockets. The ribosome contains 3 binding sites for holding tRNA. The A site accepts tRNA molecules charged with single amino acids. The P site contains the tRNA linked to the growing peptide chain, and the E, or exit site, is occupied by a tRNA molecule about to be released. The growing protein exits the ribosome through a tunnel in the large subunit. For translation to begin, the ribosome needs to be seated properly over the start codon of the mRNA, with a charged tRNA in the P site. This process of initiation, which differs significantly in prokaryotes and eukaryotes, is aided by a number of translation initiation factors. First, we will describe prokaryotic translation initiation, which requires 3 initiation factors: IF1, IF2, and IF3. Each binds the 30S small ribosomal subunit. Initiation factor 3 binds the 30S small subunit and prevents association with the 50S large subunit. This keeps the 30S small subunit open for mRNA binding. Next, initiation factor 1 nears the A site, and initiation factor 2 binds both the 30S subunit and IF1 in association with a GTP molecule. Next, both the mRNA and initiator tRNA can assemble onto the complex. Initiation factor 1 blocks tRNAs from improperly entering the A site, and IF2 brings the initiator tRNA charged with F-methionine to the P site. The ribosome binds to a sequence on the mRNA, called the Shine-Dalgarno sequence, through a complementary sequence on the 16S RNA of the small subunit. The messenger and transfer RNAs can bind the 30S subunit in either order. The small subunit of the ribosome, aligned with the mRNA start codon, is called the 30S initiation complex. The binding of the charged F-met tRNA to the small subunit initiates a sequence of events to form the 70S initiation complex. Once the F-met tRNA has bound the small subunit, IF3 falls off and is replaced by the large subunit, which completes the ribosome. Large subunit binding stimulates the GTPase activity of IF2. Specifically, the factor binding center on the large subunit stimulates IF2's GTPase activity. Because the GDP-IF2 complex has a low affinity for the ribosome, IF2 and GDP, and then IF1, are all released. This is the 70S initiation complex. Protein synthesis can now begin. In eukaryotic translation, initiation is more complex, and uses at least 8 initiation factors, compared to 3 in prokaryotes. To begin, the small ribosomal subunit binds to the 5' end of the mRNA, called a 5' cap, and then scans along the mRNA to find the start codon. Translation factors EIF3 and EIF1A bind the 40S small ribosomal subunit to prevent the association of the large 60S subunit before the mRNA can bind. Next, EIF5B recruits an EIF2 GTP MET initiator tRNA complex to the subunit, where EIF2 and EIF5B place the initiator tRNA in the P site. This forms the 43S pre-initiation complex. Note that this pre-initiation complex does not contain messenger RNA. Meanwhile, EIF4F, which consists of the 3 subunits EIF4A, EIF4E, and EIF4G, binds to the 5' end of the eukaryotic mRNA. The 4E subunit binds the 5' cap, a specialized structure at the very tip of the mRNA, while the 4A and 4G subunits bind further down. EIF4A is needed for the mRNA unwinding that will allow the ribosome to find the start codon of the mRNA. Next, EIF4B binds to the EIF4F complex and activates a helicase activity. This unwinds any secondary structure in the mRNA. The 43S pre-initiation complex can now bind to the mRNA through the interaction of EIF3 and EIF4F. Once bound, the 40S subunit scans along the message, aided by the EIF4F helicase activity, until the anticodon of the initiator MET tRNA recognizes the AUG start codon on the mRNA. EIF2 and EIF3 are released from the complex, and the 60S large subunit completes the ribosome. The remaining initiation factors are released after large subunit binding stimulates the EIF5B GTPase activity. As with prokaryotic IF2, the GTPase activity of EIF5B is activated by contact with the factor binding center on the large ribosomal subunit. The ADS initiation complex is now ready to begin protein synthesis. After an initiation complex is formed, elongation occurs. During elongation, the ribosome synthesizes proteins by linking together amino acids. 3 steps are needed to add each amino acid. A charged tRNA is placed in the A site of the ribosome. A peptide bond is formed, and the tRNA with the growing protein chain is moved from the A site to the P site.